This is the second video in a series looking at gametogenesis. In this video, we're going to be looking at the process of oogenesis, which is the production of the female gametes known as ova. This process occurs in the ovarian follicular tissue, which is found inside the ovaries. Key words associated with today's lesson are oogonium, primary oocyte, secondary oocyte, ootid, ovum and polar bodies. Now we can see from this diagram here that this process is a little bit more involved than what happens with spermatogenesis, and that is the focus of today's lesson, is walking through that process. Now, as we've said, this formation of sex cells, ova, occurs in the ovary. This is one of the ovaries supported by ligaments, and this is the oviduct or fallopian tube. Now here we have these immature egg cells which will develop and grow. Eventually the follicle that surrounds this egg gets so big that it bursts. At this point, this is ovulation, and the semi-mature egg travels down the oviduct where it may meet sperm cells and become fertilized to form a zygote. Now, you need to keep in mind that the process of oogenesis occurs in tandem with ovulation, and this will link into a later lesson. This video is a really good summary of what occurs during the process of oogenesis. I'd highly recommend that you watch it, perhaps after this video, to give you a refresher. Alright, so now we're going to be looking at how oogenesis occurs. Now remembering this is all happening in the ovaries. At the top here, similar to spermatogenesis, we have got the gamete mother cell. In this case it's an oogonium, and the plural of that is oogonia, and we know that these are diploid cells that develop into eggs eventually. So here we're giving it 2N, which of course means diploid, and then for humans that's 46 chromosomes. We have 4 single chromosomes here. Now we know that between this stage and the one directly below it, we must have mitosis, because we then have double chromosomes. So this cell has gone through interphase of the cell cycle and then mitosis. So one of these daughter cells of the oogonium becomes a primary oocyte. Now primary oocytes are also 2N, and this is the cell that starts meiosis. Now here we can see that meiosis 1 is beginning. So primary oocytes, this cell here, will start meiosis 1, but interestingly it will stop at prophase 1. So at this point here we can see that the the homologous chromosomes are crossing over here and here. Remember, the exact point of crossing over is known as chiasma. Now, this all occurs before birth in females. So oogenesis can actually have a line drawn through it here to show before birth and at puberty. So before birth we have the oogonium, which undergo mitosis and growth. We then get the first stage of meiosis occurring, which is prophase 1, and the cells are arrested at prophase 1, and they will remain in that arrested state until puberty. So when the female reaches puberty, what we then have happening is the primary oocyte will continue its maturation. The first strand of meiosis will run to completion, and we can see here that we have the haploid number of chromosomes in the resulting cells. So the primary oocyte at the onset of puberty will complete its first strand of meiosis, and we get 2 cells forming. The first one is known as a secondary oocyte, Now, it has the double chromosomes, but it has the haploid number. So that's 23 chromosomes here, just represented as 2. The other cell is known as a polar body. Now, you can see that the polar body is much smaller than the secondary oocyte. And the reason for this difference is because the secondary oocyte gets more of the chromosomes, more of the cytoplasm, but half the chromosomes. The polar body receives half the chromosomes again, but hardly any of the cytoplasm. So really, polar bodies are just a mechanism of getting rid of the excess chromosomes, because remember, in meiosis, we need to have single haploid cells. Now, as the secondary oocyte gears up for another round of meiosis, so meiosis 2, it will start that process, and it will form this cell here, which is an ootid. An ootid is an immature egg. Now, in order to get rid of the other chromosomes that it doesn't need, because remember, we need to have single chromosomes that are haploid, it produces another polar body. Now, this ootid, produced from the secondary oocyte, will eventually become a mature ovum. Now, the mature ovum only forms when meiosis 2 is finished. Now, meiosis 2 will only actually complete its full run if this ovum becomes fertilized by a sperm. Now, that might seem a bit odd, but it doesn't need to complete its full meiotic cycle if it's not going to become anything. So it doesn't really need to do that. So what we have at the start, we have the oogonium. It turns into a primary oocyte. We stop here until puberty. And then at puberty, meiosis restarts, and we have the first round of meiosis complete. We produce a secondary oocyte and a polar body. Second round of meiosis occurs, and we get a secondary oocyte becoming an ootid and another polar body. The ootid, which is an immature egg, will become mature and complete the rest of meiosis only if fertilization occurs. So like any type of meiosis, we start off with a diploid cell, one diploid cell, that eventually becomes 4 non-identical daughter cells. But in the case of oogenesis, only one of those daughter cells is a viable egg, and the rest are polar bodies. Now, these polar bodies will eventually disintegrate, and they're not used for anything. In some cases, they can be fertilized, but this is just the exception rather than the rule. Okay, so this diagram can be found on page 10 of your class booklet. I've completed this to help you fill this out, but I would like you to annotate this diagram using the previous slide to help you understand the process. So again, we have one oogonium. It goes through growth and mitosis. We end up with a primary oocyte. This primary oocyte is formed in the womb before birth, and we only go through prophase 1 of meiosis 1 here. At puberty, meiosis 1 restarts and is completed, and we end up with a secondary oocyte and the first polar body. Polar bodies are smaller because they have much less cytoplasm, but they still contain half of the chromosomal information. Sometimes the first polar body can split into 2 other polar bodies, which accounts for the 3 polar bodies. And the secondary oocyte will start meiosis 2, but then it will stop at metaphase 2. At ovulation, this ootid will maybe become fertilized, and if it does, metaphase 2 will be complete, anaphase and telophase and so on, and then we will end up with a mature ovum. The mature ovum, of course, will then turn into a zygote if fertilization has occurred. And then we have our second polar body, or maybe third, depending on how you count. Okay, so that is the process of oogenesis. This is a little image to show you the differences between the two. If you're more of a visual person, if you prefer that in a table, this is a good comparison just to help you understand the process in terms of the location, the number of gametes produced, and the timing and the onset of those processes. Okay, so to help you again with your booklet, if you turn to page 11 of your booklet, I'm just going to summarize what happens during spermatogenesis and oogenesis. Remember, tables are a really good way of summarizing your knowledge, and they can be used effectively in extended responses. So let's look at spermatogenesis first. The location this takes place in, remembering that we need to be specific, it is the seminiferous tubules in the testes. You wouldn't get any marks if you just said testes, you have to say exactly where. The mother cells are the spermatogonia, and this process, of course, only starts at puberty, but from this point on, it is continuous until death. The immature spermatozoa will reach their final maturity 72 days later. This, of course, happens in the epididymis. Now, the final number of cells that are formed are 4. These cells are haploid spermatids, and they will form from secondary spermatocytes. Now, the other products that are formed during spermatogenesis are the seminal fluid, which is the liquid that sperms swim in. This is formed elsewhere in the body, namely through the seminal vesicles, the prostate gland, and a little bit of contribution from the bulbourethral gland. Okay, so now looking at oogenesis, this takes place in the ovaries, of course, and the mother cells are known as oogonium. Now, very different to spermatogenesis, this series of events occurs before birth, where we give rise to primary oocytes. These primary oocytes are arrested in their development that stops at prophase 1 of meiosis 1. Okay, the next stage in the cycle restarts at puberty. The prophase 1 will restart, and the primary follicle and therefore the primary oocyte will develop into a secondary follicle containing the secondary oocyte. The final cells that are produced are the ootid, which is an immature egg. Now, meiosis is not completed at this point. Remember, we are arrested in metaphase 2, but we do have an immature egg and 3 polar bodies. The other products formed apart from the main one, which is the ootid or the egg, are the first and second polar bodies. Now remember, we refer to 3 polar bodies being formed because the first one will often split into 2. Okay, so that is a summary of both spermatogenesis and oogenesis. This video here is definitely worth a watch. It's only about 3 minutes long, so please take the time to watch it and really consolidate your knowledge. Before you finish up today, please ensure that pages 10 and 11 of your class booklet are complete, and ensure that you have read and understand the content on pages 283 to 284 of your textbook.